In this lecture, we're going to continue our examination of how sounds are specifically transmitted at different locations throughout the cochlea. And when we take a look at cochlear anatomy, the structure of that organ narrows from the base to the apex. And in this somewhat illustration here, I try to illustrate how that base does the narrowing of that portion of the cochlea if we roll it out. On the next slide will be a more accurate picture. Um, but now the basilar membrane widens towards the apex and becomes thinner. So in this illustration here, this is my attempt to illustrate the basilar membrane, where the left-hand side represents the base of the cochlea, and the location on the right-hand side represents the apex. And you can see how the basilar membrane in this picture here, anyways, appears to be thinner but wider. And these two waveforms here represent two different sound transmissions. And because of the shape of the basilar membrane, this allows what's called frequency separation. The frequency separation is referred to as tuning. And we see that here with the difference between a high frequency wave that tends to be detected much earlier, at least in terms of the anatomy of the cochlea, and the low frequency waves that take longer to transmit one waveform. And that tends to be detected at the more distal parts of the cochlea towards the apex. And we see that a little bit clearer here on this picture. If we look at the top left-hand corner, we have our cochlea, our spiral organ, that if we roll it out, we have this long structure. And on the right-hand side, we can see the different parts of the basilar membrane as it is wider and thinner towards the apex as opposed to the base. Once again here, pointing out to the ossicles, the inner ear bones, that will transduce the mechanical reverberation of the tympanic membrane into the oval window at the base of the cochlea. So we have our cochlear base, we have our cochlear apex once again. And when we talk about afferent sound transmission, the sound transmission that will eventually be converted to bioelectrical signals are conveyed mostly by the inner hair cells. So over 90% of the sound that gets transmitted into neural signals comes through the displacement of the inner hair cells. And there are an estimated 3,500 of these inner hair cells. And what we see here on the bottom are four different sample sound waveforms. And so here we see a very high frequency a high frequency waveform characterized at 1600 hertz. Another one not quite as high, but still considered fairly high at 800 hertz. And on the right-hand side, we have a 50 hertz and 25 hertz, so lower frequencies. And I'm using the boxes here, the color of the boxes, to indicate where those sound waves would likely get transmitted into bioelectrical signals. And so the blue boxes here point to the high frequency waves, which would tend to be detected near the cochlear base, whereas the much longer waveforms would not be detected until those waveforms cause a mechanical disturbance, a waveform, if you will, through of the basilar membrane out towards the cochlear apex, hence the red boxes pointing to this red colored region of the unrolled cochlea. And so now the next question that comes up is, what about the outer hair cells? We know the outer hair cells are much larger in number than the inner hair cells, but the outer hair cells are quite interesting because they have what are called efferent connections from the brain. So the afferent, which transduces sound pressure waves into bioelectrical signals, travels towards the brain. We have bioelectrical signals traveling from the brain towards the outer hair cells. And so that conveyance will influence how the afferent, you can see up here when I talk about the afferent, how those signals are conveyed. So the brain does play a role in tuning the transmission of the sound waves by the inner hair cells. And to understand that, we go to a little bit more detail on the auditory nerve. We know the auditory nerve is the nerve that conducts the bioelectrical impulses towards the brain, where we eventually hear the sounds. Once again, hearing is the perception of these pressure waves. And so, as indicated on the previous slide, most of the neurons that carry that information come from the inner hair cells, so over 90% come from those cells. And the nerve is made up of a very large number of fibers or neurons. So the term fiber, you can see this here on these pictures, and neuron are used interchangeably. So different auditory neurons will respond to different sound frequencies. So not only do we have, okay, I'm making reference to the previous slide, not only do we have the localization of sound frequency through the cochlea, but we also have the individual neurons that operate within a restricted range. Each neuron is specifically tuned to transmit bioelectrical signals in response to a particular frequency. And so with these are four examples of different fibers. They're named according to, I skipped a few fibers here, but these represent four different neurons of the auditory nerve that transmit bioelectrical signals. And these are on the order of kilohertz, so 1,000 hertz, 10,000 hertz, 100 hertz, as you can see here along the x-axis. And the threshold intensity refers to the intensity of the sound wave that will generate an impulse, a bioelectrical signal to be transmitted along those nerve fibers. And if we locate the portion along the x-axis, if we just compare fibers 1 and 2, we see that fiber 1 will see this very steep increase in discharge at a lower frequency than fiber number 2. So fiber number 2 doesn't immediately start to increase its transmission until a higher frequency wave shows up. Now, when we look at fibers 5 and 6, we see similar patterns, but the high discharge comes at much higher frequencies. And because these are different neurons, each neuron is tuned, if you will, to detecting and being sensitive to a particular mechanical frequency of those pressure waves. On this slide, we're going to take a look at the outer hair cells. So recall that the inner hair cells in this computer-enhanced electron micrograph at the top right-hand corner, here you see computer-colored yellow are the inner hair cells, and the color for the of green for the outer hair cells. Once again, the outer hair cells far outnumber the inner hair cells, yet the inner hair cells are the ones responsible for the transmission, largely responsible for the transmission of pressure waves into bioelectrical signals, and you can see them embedded here in the tectorial membrane. So the outer hair cells play an important role in that it helps to tune the inner hair cells. This depends on the outer hair cells. So it gives a degree of sharpness, and that sharpness will make the inner hair cells even more finely tuned, to the pressure wave coming through the basilar membrane that will eventually cause the shear in the tectorial membrane. And the mechanism is referred to as electromotility. And electromotility refers to the lengthening and the contracting of the hair cells. And this occurs after action potential generation. So if we take a look at this picture here on the bottom, here we have our auditory nerve. And the auditory nerve has obviously connections to the inner hair cells. That's this first, the narrow red box, pointing to this portion of the tectorial membrane. And this will transmit afferent information. You can see here afferent information going out towards the brain. Yet the big red box here, pointing to the outer hair cells projecting into the tectorial membrane, are innervated by efferent fibers. I don't have a box around this, but these are the efferent fibers, which are transmitting action potentials in the direction of the outer hair cells. So the outer hair cells, as you can see, both in the upper right-hand electron micrograph, the bottom left-hand picture here, I think conveyed a little bit better here on the top right-hand corner, where you see that these greens are projecting a little bit further into the tectorial membrane than the yellow ones. So they do extend further, and in doing so, when these cells are activated efferently, they will tend to make the cochlear partition more stiff. Now, that does not mean that the cochlear partition will not move and not create shear. It does, but a stiffer cochlear partition will make the cells, make the inner hair cells and the tectorial membrane more sensitive to the transmission of those mechanical waves. So the inner hair cells do become more sensitive to those fine types of frequencies. And so that's what allows a sharper tuning between different sound waves that have slightly different frequencies. Another illustration here in this bottom figure will depict where these inner hair fibers are tuned depending upon the activation of the outer hair cells. So if we just, in the absence of a mechanical wave, which is the actual sound being transmitted, um, will outer hair cell activation, so with no sound, if we were to send an efferent signal to the outer hair cells, what would basically happen here? That will actually cause an action potential of the inner hair cells, and that's called otoacoustic emission. The otoacoustic emission is where the external hair cell contraction will immediately discharge action potentials. And this technique is often used to diagnose hearing ability. And on the picture here, we'll illustrate the impact of the outer hair cell. So here we see a range of different frequencies. We've got three different fibers here, fiber 2, 4, and 6, color-coded. And so in the light blue, dark blue, and the red. And you can see that these three particular neurons are tuned to different frequencies. And so what we're tracking here is something we saw on slide number 7. And this is the threshold intensity that needs to activate the fiber above its what's called a spontaneous firing rate. So we just take a look at where we see this very steep increase in the red, very steep increase in the light blue, very steep increase in the dark blue, and those occur at different frequencies. And so these are the inner hair cells. So the inner hair cells are considered neurons. They don't look like neurons, but they are considered neurons. Now the dashed curve here reflects, let me just put that arrow here, the dashed line represents the threshold intensity of the fibers, So of the fibers, when the outer hair cell is not activated by the brain. So if we have no efferent fibers, then these three sharp tuning responses would collapse into the dotted line here. showing that without the stiffening of the tectorial membrane, the ability to differentiate on a very fine basis sound waves of different frequencies will not be possible. So not only is the tectorial membrane uh, finely tuned to the transmission of pressure waves, it's not good enough. We need the efferent connection to the outer hair cells to create even more stiffening, giving rise to a more acute differentiation of pressure waves of different frequencies. So this ends our video here on part 2 of the auditory